Okay, if I could have everyone's attention, we're going to get started today. So thank you all very much for coming today. Welcome to today's sustainability seminar. My name is Nancy Holm. I'm Assistant Director here at the Illinois Sustainable Technology Center. Thanks for making the drive in and around to get to our seminar today. And I organize the seminars along with Beth Mishesky here at ISTC. Our seminar series, as those of you may know who attended the last one, are focusing on water quality and water conservation this semester. Each semester we have a theme for our seminars, and we have over 140 seminars online archived there from the past 8 years on a wide variety of sustainability topics for those who might be interested in looking back at those. The schedule for the seminars for this semester are on the table outside the conference room here and also online under events on our website. In case you can't attend the seminars in person, we do broadcast them live, and then as I mentioned, we do archive them on our website. It usually takes a few days to a week to get that up after the talk. So I would like everyone now to please, if you haven't already done so, silent your cell phones. And then also we'll hold all the questions for the speaker till the end. And then also for those online listening, you can type your questions in at any time, and then we'll have those at the end also. I'm very pleased today to introduce a colleague of mine, Dr. Wei Zheng. Dr. Zheng joined ISTC in 2008 as a senior research chemist, and he's also an adjunct faculty member at the Department of Natural Resources and Environmental Science here at the U of I in Champaign, Urbana. Prior to joining ISTC, he worked as a research scientist at the USDA Agricultural Research Service in their salinity lab, and then also at the University of California in Riverside, California. He earned his PhD in environmental chemistry at Zheng University in China. Currently, Dr. Zheng's research is focused on emerging contaminants and pathogens associated with reclaimed water use and wastewater treatment, on environmental fate and pollution prevention of pesticides, on biochar production and application, and also on post-combustion carbon capture. He's associate editor for the Journal of Environmental Quality, serves on the editorial board for the International Journal of Environmental Monitoring and Analysis, and is on several review panels for the National Science Foundation and USDA. Today, Dr. Zheng will be discussing his projects on pharmaceuticals and personal care products in the environment. I'd also like to mention that in the spring, ISTC will be hosting a conference, and there's flyers about this out on the table, or for those listening online, we will have information up on our website. But on April 4th at the iHotel, we'll be having a conference dedicated to pharmaceuticals and personal care products in the environment. We'll have several tracks on different research aspects as well as education related to this topic. So we hope you will all watch for that. We'll have a call for abstracts in December and have more information on our website. So without further ado, I'll introduce Dr. Zheng for his talk. Thanks.
Thanks, Nancy. Good afternoon. First of all, I want to thank you, all of you, come here to ISTC, discussing and exchanging your interest on the PPCP works. Today, I would like to talk about some of our ISTC works on emerging contaminants. In this semester, since the seminar, the topic is about water quality and water conservation. So today my talk mainly focuses on emerging contaminant issue in the water. Currently, a drought is a big challenge for us. Water shortage is a top global issue. Currently, more than 1 in every 6 people in the world doesn't have access to potable water. In the U.S., some states like California, in the last couple of years, is experiencing extreme and exceptional drought. So it's hurting their more than $46 billion agriculture industry. Also, even in the Illinois, we thought we Usually we have a lot of water. But in the summer 2012, the USDA lists 19A of Illinois countries as a primary natural disaster area because of the drought. So water reuse is one of our primary solutions to mitigate the drought. Using treated wastewater for the land application can provide nutrients and organic matter for plant growth and reduce high quality water using and save the valuable water, fresh water. Currently, there are three primary types of wastewater for reuse. One is treated industry or municipal sewage wastewater. Second is treated domestic wastewater. Third is wastewater from concentrated animal feeding operation system, like large animal farms. This classify based on my research work, the purpose. So, in the big city, we have a large sewage treatment plant to clean up our generate wastewater. The treated wastewater could be used for land-scale irrigation. Also, if we're using some advanced treatment technique, we can convert the toilet water to top water. For example, California Orange County Water District, they combine the microfiltration, reverse osmosis, and the UV light. They can purify the toilet water for drinking. Even using some advanced technique, the NASA, they can purify our urine to drinking water. So next time if we want to travel to the Mars, we can drink our generated water. Also like this beer garage, we can drink our urine water and show our president how to survive in the wilderness. In the small town, aerated lagoon had been used to treat domestic water for many years. In the U.S., right now about 8,000 such this waste treatment lagoon. In the large animal farm, like dairy farm, swine farm, they generate large manure and manure contaminated wastewater. Typically, this wastewater can be used as a fertilizer for agriculture land application. So this wastewater, even after treatment, they still have a lot of contaminants. For example, they have like a conventional contaminant, this common contaminants, like excess amounts of nutrients, salts, microorganisms like passage, heavy metals, and pesticides. Also, this wastewater may have some emerging contaminants, like pharmaceutical and personal care products, natural hormones, PPDs, PFCs. So for my research, right now, most focus on the PPCP and the natural hormone, these emerging contaminants. Currently, PPCP are receiving great public attention because they had to be frequently detected in the lake, river, and stream in the United States. The occurrence of the PPCP in groundwater and surface water will definitely impact water quality. For example, some PPCP shows very highly acute toxicity to aquatic species. Some PPCP have a potential to change watershed ecological system. But their long-term effect on human health, we don't know. Also, because they may be a chronic compound. Some still, most still like hormone are one of important classes of emerging contaminants. They are either natural or synthetic compounds. So if you want to build a muscle like their guys, you need to really work out. Don't use any hormone. Because most steroid hormone are classified as highly potent endocrine disrupting chemicals. they can interfere with the normal function of endocrine disrupting system. But their long-term effect on human health are unknown. But in the United States, the opposite is often suspected to relate too much consumption of human growth hormones. In the aquatic environments, still high hormone, even at very low concentration, like a nanogram per liter, still can adversely affect the reproduction of freshwater species. For example, some hormones can induce male fish feminization. Also, you heard about some interesting story. People find a frog with 2 or 3 heads. This is because the living water may be contaminated by these endocrine disrupting chemicals. So compared to the conventional contaminants, the emerging contaminants have 3 different features. One is the most emerging contaminants are not acute toxic at their environmental concentration, but most of them may chronic. Also, their long-term effect on the human health unknown. Second, the concentration for this emerging contaminant in the aquatic environment is very low compared to the convention. For the convention contaminants, their concentration usually in the environment ranging from the ppb to ppm level, like a parts per billion to parts per million. But for the emerging contaminant in the environment, the often happen like a ppt to ppb, the parts per trillion to parts per billion. It's kind of like a nanogram per liter to milligram per liter, even lower. How low for this, the concentration? Maybe I using money to demonstrate. You'll be easy to understand. So what's this? This is 1 cent, right? So 1 ppm, like 1 parts per million, kind of like a 1 cent reduction from $10,000 transaction. It's kind of like ask you find this a penny from 1 million pennies. Can you? 1 ppb kind of like a 1 cent reduction from $10 million. It's kind of like ask you to find a needle in a haystack. 1 ppt, the parts per trillion. You can see, you can imagine. I'm a rich guy. I have $10 billion. So I can buy the most 100 times like a most expensive house. This house area kind of like big as a 4 big city. Right now, in this big city, I want to find 1 cent. Can you? So compared to the conventional contaminants, the third future for emerging contaminants is regulation missing. Because of lacking information and the knowledge of emerging contaminants or their presence, fate, and effect in the environment, state and the federal government agencies, they cannot widely and properly manage and regulate this compound. So this raises a lot of questions we need to answer. For example, like what kind of emerging contaminant in the environment, where they come from, what environment fate and effector, and what pollution prevention strategy to minimize this emerging contaminant loading in the environment, and for contaminated water, what kind of treatment technique we can use to remove them from the water, remove them. So thanks Nancy and Beth. So from our website, you can clearly see what our ISTC working on some PPCP projects. Here I mentioned a little bit of our study. First is an ongoing project. This project is funded by University of Illinois Extension and Outreach Initiative. So we work in this project. We're working with Illinois Sea Grant, Laura Cam, and Professor Michael Prower from the Department of Corporate Science. So we want to try to solve some PPCP issue in Illinois. In this project including 3 components: research, education, and outreach. Through the extension initiative, we try to provide some science-based PPCP information and pollution prevention strategies to the communities. Second project was funded by NGG REC. In this project, 4 scientists for 4 surveys, like Illinois Natural History Survey, Illinois State Water Survey, Illinois Geology Survey, and RS, to create the interdisciplinary team. So in this project, we want to analyze or monitor karst groundwater contaminants in the western Illinois. Also, the third project is a similar second project, is funded from our institute. This is PRR MAP funds. In this project, we want monitoring detect PPCP in karst groundwater in southwestern Illinois. Fourth project is funded by Illinois Hazardous Research Funds. This project is to monitor the PPCP and steroid hormones in the municipal sewage treatment plant and evaluate different treatment stages, how to remove this PPCP. Fifth project is to investigate the FEED PPCP in the irrigate wastewater effluent. This project is a work with Illinois State University. Currently in the city of Lexington, they are using the treated wastewater to irrigate agricultural farm. So in this project, my role is to investigate the environmental fit of the PPCP, also assess their potential impact on the surrounding watershed. Sixth project is to invest uptake and accumulation of PPCPs and hormones in the vegetable plant. They are glued in the contaminated water. In this project, we want to see if use the agricultural irrigation, agricultural irrigation with the PPCP and the hormone contaminated water could be or not a food safety problem. The seventh project is funded by USDA watershed program. In this project, we want to understand if the hormone and the antibiotics they release to environment, what's their environment fate and the transport, how they are leaching to groundwater, how run off to surface water. So this project, you can see, they mostly involve the laboratory, field, and the modeling experiment to monitor occurrence of emerging contaminant in the environment, to demonstrate their environment fate and transport, and develop effective management strategy and the treatment technique to minimize, this emerging contaminant loading to the environment and prevent their pollution. So in here, based on this project, I'm mainly talking about the 4 research activity and some results. First is for the method development. There are 2 challenges for the method development. First is the complex matrix. You can see some wastewater sample very dirty. So we developed some isotopic dilution method to reduce matrix effect for the LC tandem mass and GC mass analysis. Second, as I mentioned, most emerging compound in the water sample, the concentration is very low from the PPT to PPB level. So we need to develop a method can analyze, you have a low detection limit. This slide shows how we prepare the samples. After we collect the sample, we spike the isotope compound and then filter. We use the SPE, solid phase extraction, to extract the emerging contaminants. After concentrated extract, we're using advanced analytic tech instrument, GC mass and LC tandem mass to analyze. So use this method, we can analyze the most emerging contaminant at a nanogram per liter level. So currently our lab have a capacity to analyze simultaneously analyze more than 60 PPCP and over 10 steroid hormones. This table shows our the master LD, the master limit of detection limit for some our PPCP and the hormones. You can see this for most the target compound, that analysis detection limit can as low as nanogram per liter. Also, they have a very good recovery. Based on the developed method, we go into the monitoring occurrence of emerging contaminant and identify the pollution source and determine how they are reaching to the, how they are released to the environment. This is one of our project. In one of our project, we collect some spring and cave water from the Illinois karst ground area. After analyzing this water sample, we detect several PPCPs in this karst groundwater. Their concentration very low, around like a nanogram per liter level. Maybe you still doubt why, where this compound come from, because this karst groundwater, this groundwater usually is our drinking water supplies. So we know that most emerging contaminants like PPCP hormones, they originally come from the human and animal exclusion, also from the waste disposal. So that's why I want to remind everyone, we don't want to just dump our unused medicine in the toilet and the trash. Also, don't like this guy pee in the river, point our, drinking water supplies. As I mentioned, emerging contaminants, they are being loaded to the environment through three main ways. It's discharging from the municipal sewage treatment plant, discharging from sewage treatment lagoon, and also from the wastewater use for the large animal farm. Usually, emerging contaminant in the wastewater cannot be completely removed by sewage treatment plant. Effluent discharge, we are loading undegraded emerging contaminant to the surrounding environment. So that's why we have a project to investigate the monitor occurrence emerging contaminant in the municipal sewage treatment plant. Also, we want to assess the removed efficiency through different treatment the system. First, in this project, this is the season sampling and the monitoring project. After we collect the sample from each treatment sites, for analysis, then send to the lab for analysis. This slide I show some results. For the influent, we saw that we detect over 13 PPCPs, but their concentration ranging from the PPB to ppm level. After 3-stage treatment, their concentration largely reduced. Some PPCP we cannot detect. Even detect the PPCP, their concentration ranging reduced to the PPB, PPT to PPB level. Also we do another project, working on the occurrence of emerging contaminant in sewage treatment lagoon. This is a typical Illinois, sewage treatment lagoon for the small town, for small community. The wastewater from the small town will pump to the 2 aerated lagoon. After this aerated lagoon, then filtered by the center tank. After filtration, this treat the effluent direct discharging to surround the creek. Usually, this procedure takes 3 days from influent to effluent. So we collect the water sample from each aerated lagoon for the analysis. You can see for the influent, we detect about 10 PPCPs in the influent. Their concentration ranging from the PPB to ppm level. After 2 aerating lagoon treatment, except carbamazepine, most compounds, the concentration largely reduced. In the influent, the concentration ranging ranging from the PPT to PPB level. Except we analyze this lagoon system, also we collect some creek upstream and downstream and the river water. In this, we want to see if discharging treated sewage lagoon waste can infect surrounding watershed. Here are some results. We find, in the upstream, we detect several PPCP. Their concentration close our detection, the mass detection limit. But for the influent, we can see their concentration like 1,000, 100 times higher than background, like upstream. After effluent discharging to the creek, we found a high concentration the PPCP in the downstream creek. Is this even 2 miles from the effluent? This creek runs to the Mexican River. So we also detect these emerging contaminants in the river. But they are because larger dilution, their concentration can around like 1 ppm to the 1 ppt to 10 ppt, 1 to 10 like a nanogram per liter. So this study suggestion, direct discharging, the even treated lagoon water still may, significantly elevate surrounding groundwater, the PPCP concentration. Also, we invested another emerging contaminant source, like a large animal farm, CAFOs. This is a typical, California daily fund wastewater profile system. So most water generated from milk and parlour. If you generate 1 gallon milk, you need 1,000 gallon water. So wastewater fresh from milk parlour through the sewage land and run to the lagoon and stored in the lagoon. This lagoon water usually using for the applied to the surrounding the field for the nutrient purpose. So then to grow this field grow the alfalfa feed to the cow. So following this wastewater follow, we sequentially take the wastewater sample for analysis. We find the total concentration of steroid hormone decrease around this daily, the wastewater follow. Because, the dilution in this system, dilution, biodegradation, photodegradation, sorption, and the setting of hormone-associated manure particle will largely reduce the steroid hormone in this system. So after this system analysis, we propose some management strategy to try to help the farmer help us to minimize their hormone loading to environment. For example, we're using sequencing the lagoon, increase the residence time of wastewater in lagoons, increasing the piling time of solid waste can significantly reduce, or minimize this lagoon, the hormone contaminant loading to the environment. Also, my third main research activity is we want to understand if this emerging contaminant enter to the environment, what is their environment feed and transport. Here I show you some results. This slide shows some degradation connects our steroid hormone under different conditions. You can say usually under anaerobic condition, we find the steroid hormone, the degradation is much faster than anaerobic condition. After analyze their degradation process and identify degradation products, we proposed some degradation pathway. Most interesting, we find this reverse reaction among the 3 hormones. That means these 3 hormones under anaerobic condition. So this means in these 3 hormones, because this reverse reaction, they not degrade. So that's why we suggest anaerobic condition may inhibit reduction of steroid hormone. So this causes their accumulation in the anoxic environment, like sediment and the groundwater. Also, this is reverse reaction involved 2 reverse first order reaction. So we're using some reaction model to calculate the degradation rates. Following the degradation study, we also conduct a soil column experiment, try to demonstrate how the emerging contaminant, the movement in the soil. So this have a sample. This is for the sample for the antibiotics. Fluorophenical. Fluorophenical is a veterinary antibiotics. This slide shows the transport of our fluorophenical in the compared to the artificial rainwater. The transport of the fluorophenical and the dilute, the daily wastewater, the transport facilitated. This because then based on this study, we suggest some mechanism. We know that most fluorophenical, most soil, their surface negative charge. Chronic, you can mention the chronic is kind of dissolved organic matter. They are negative charge. So chronic can quickly move through the soil. So that's why we proposed why the fluorophenical can quickly transport in the soil. One is the mechanism is from the competition absorption. Second is chloride can absorb fluoronic and carry it over. Third is the chloride can dislodge, absorb the fluorophenical and the chloride carry it over. So also we developed a model to simulate the facilitated transport processor. Using our model, we can very good fit our transportation data. Another, we do the field monitoring. We collect a sample from the field that receiving some CAFOs wastewater or manure. We detect some steroid hormones from the surrounding groundwater. So finally, I will mention some mitigation strategy. This including like a pollution prevention strategy. Also for we want to use this strategy. Want to prevent, minimize the emerging contaminant loading in the environment. Also for contaminated water, we want to develop some innovative treatment technique. We can efficient and cost effect to remove this emerging contaminant. For the pharmaceutical waste, the best way to deal is not create in the first place. What's this mean? This means that we not dump any medicine to the toilet and the trash. We can correct this medicine, unused medicine. So that's why in our UVA extension grant, we're working with Laura Cameron from the C grant because they have a medicine collection program. In this program, they set up a medicine collection box in the police office. So you can drop the medicine in like every day, 24 hours per day, 7 days per week. So, but also very important, this is free. In this medicine take back program, they already have like 72 communities in Illinois, Indiana, Wisconsin, and Michigan to ensure the properly dispose more than like 50 tons of medication. Also for contaminated water, we try to develop some efficient or cost-effective method to remove them. In one of our project, we're using vegetable oil to remove emerging contaminants. Compared to the mostly typically using the method like active carbon, this vegetable oil treatment have 3 advantages. First, in our Midwest, like corn oil is kind of cheap than the active carbon. Second is because most of emerging contaminants, they are hydrophobic. So the use the vegetable oil can very efficiently remove this contaminant. Third, after you extraction, the contaminants, the oil, we can make the biodiesel or burning, still use energy. Not like active carbon, you need a land disposal. So here we show you some study. We test the different vegetable oil. We find the corn oil. You can very efficient to remove some steroid hormone. Even the remove efficiency can up to the 100% if you depend on the system. Also, we're working with Dr. Wensu Liu. He is a professor from the Emmanuel Civil Engineering. He developed artificial riverbed, the filtration to remove some nutrients. But in this one, we want to use this technique to remove emerging contaminant from point and non-point source. I think this technique have a potential to remove large scale the contaminated water. Also, I worked with Dr. Cai Xingyue to develop some cyclodextrin and biochar-based nanomaterial to selectively capture trace level emerging contaminant or discharge absorption the contaminant and regenerate the solvent. This technique have 2 advantages compared to the active carbon. One is, this can using this. I mentioned most waste, the emerging contaminant concentration in the water is very low, because another contaminant like dissolved organic matter or some common contaminant will compete absorption if you use the biochar, if you use the active carbon. But we use the cyclodextrin can selectively capture this contaminant. Also use this material. Second advantage is they can easy to regenerate, so we can reuse this solvent. So, but the disadvantage for cyclodextrin is because this is water soluble, it's difficult to operate. So that's why we combine the technical, the cyclodextrin and our biochar research. We want to coating this compound to the biochar, so to try to remove some low concentration, like trace concentration emerging contaminants. So if for all the remove emerging contaminants, this is not only water involved, also relate to the food and the water and energy. If you want, so that's why any treatment, you need a lot of energy, right? If whatever you use the membrane, whatever or another system, you need the energy. So we need to develop the innovative technique, use less energy to remove, get a high quality water. Also, sometimes we need to consider what kind of widely using the water. We need using right water for right place in the right time. For example, for the fresh products before the harvest, maybe we need to use high quality water because of the food safety concern. Okay, before close my talking, I give you quickly review. Currently, jaw is a big challenge for us. Water reuse is one of our primary solutions to mitigate the jaw. Using treated wastewater for land application can provide nutrients and reduce high quality water use. This water can, even after treatment, still have a lot of contaminants. For my research, most focus on the emerging contaminants like PPCP and natural hormones. In this talk, I present 4 main research activities and the results we are conducted. First is method development. We develop several methods to analyze the PPCP and hormones with the detection limit at the nanogram per liter level. Also, based on the developed method, we monitor the occurrence of emerging contaminant in the municipal sewage treatment plant and in sewage treatment lagoons and also in the large animal farms. Third part, we also investigate the feed and environment feed and transport for emerging contaminants once they are come to the environment. For the finally, I mentioned that we developed several the mitigation strategy. For example, we're using a medicine take-back program. We can prevent the contaminant, emerging contaminant loading to the environment. Also, for the contaminant water, we can using for the vegetable oil extraction. Also using like artificial river band system and like a cyclodoctrine and bio-based absorption to remove the emerging contaminant from the water. Finally, I will thank you, my collaborate, my colleague, and the people working these projects. But also, I want to appreciate all the funding agencies support this research. Now I'll take your question. Also, I'm like any collaboration, looking for any collaboration. Thank you. So when we show,
this moment,
maybe
wait, it's not working.
Oh, I'm sorry, I didn't press this one. Yeah.
Okay. We have a little battery problem. Just one moment.
No, I could be way off.
Okay, so thank you very much, Wei. What we'll do now is take questions. I'll bring the microphone around to the audience here so that we can get those on our tape that we're making of this. And then we'll take some questions from those who may have typed them in online. So, Dr. Pova?
So Wei, thank you very much for a very good lecture. The question I have is, when you show the degradation of some of these contaminants, what do they degrade into, and could some of the degradation products be more toxic than the original parent compounds?
Yeah, it's a great question. Yes, this is what we did. For example, we do some the estrogen hormone for degradation. So we find, if you depend on what kind of degradation the condition. So for example, like aerobic condition, they will be quickly degrade. We can monitor some degradation product, but in the anaerobic condition, some degradation products will, may be more toxic. For example, 17-beta estradiol to 17-alpha estradiol, their degradation, the toxicity is different. So that's why I didn't do this toxicity analysis, but we just do chemical concentration. But we know because this compound, the toxicity, a lot of people already did, so that's why we said, this reaction maybe sometimes toxicity will increase for the degradation product.
Are there other questions here in the audience? If not, are there some online?
Could you go back to the slide where you had the following the vegetable oil extraction? You went through that pretty quickly. And
this one?
I was curious, were there some of the compounds that were more preferentially extracted by the vegetable oil than others?
Yes, it's good. Yeah. This is most, the emerging contaminant, the hydrophobic. For some hydrophobic PPCP, maybe not very efficiently removed by the vegetable oil. So that's why we concerned about maybe sometimes we modify. We do some simple modify some vegetable oil, we make them more hydrophilic. Maybe still can remove. Sometimes for example, you just like a surfactant, maybe have a two-phase, you can move both. So that's why we need something we need to modify. We're still working project, so we want to modify some vegetable oil. Yes. Yes. This is because for the the hormone compound like this kind of like a triclosan, they are very very hydrophobic compound, so they can easily removed by vegetable oil.
Other questions?
Yeah, actually, I got two. The first one is, with all the stuff you're doing, how the heck do you have time for any more collaboration? That's the first question. But the second one, more serious, is what about temperature? Degradation. It gets cold here, and there's got to be some effect on that and how fast these things degrade or persist or whatever.
Thanks for the question, for the collaboration. So that's why I work in different projects, working with different people, using different expertise to working together. So I appreciate everyone, here I appreciate all my collaborate. So for the second question, we did do different, for the degradation, we do different temperature. Usually in California, there's a warm area. In the Illinois, maybe the winter time have a cold. So that's why we do different temperature. Usually, most like a 30 degree to 40 degree is very good to remove to degradation. But maybe low temperature sometimes will inhibit degradation. So that's why in the winter time, so more persistent. So that's why we do, this time we want to say, for the cast groundwater, we expect in the winter time, maybe more PPCP or hormone accumulate in the water.
In one of your recommendations, you suggest the use of multiple stage of lagoons. But the actual practice is now we are getting rid of lagoons. So in your opinions, how can we expedite the degradations instead of multi-stage lagoons?
It's a good question. This is because some, I think it depends the daily farm, animal farm system. In California, they most using multiple lagoon. The purpose they install the wastewater because you need animal wastewater because this depends the system. If you have a large scale, you need a different lagoon to do this. So, but I consider about the Illinois, maybe some small, not a big, small, not like a small animal farm. They just one lagoon. But this is why we saw that if you use the multiple, sequential multiple lagoon, will help to the degradation. So help the, contaminants set down. So that's why we saw that we just recommend this technique, this way maybe can help to reduce or minimize the hormones contaminant, later for the irrigation if landed to the environment.
You got an online question, Mark. Just read it off.
You can read it off.
Yeah, you can just read it and start answering.
This question about the carbamazepine concentration increase during the wastewater treatment. Was that increase statistically significant? If so, do you have a rationale? We don't do statistical analysis for this, but carbamazepine concentration increase because this compound not easy to remove in the sewage treatment plant. So that's why they can increase. Also, this is usually using the carbamazepine used for the cheese market.
Yes, can you comment on the legislative landscape for discharge of these compounds or drinking water? I mean, we're talking about, removal, but is there anything forcing it at this point?
Okay. So as I mentioned, most like emerging contaminants like PPCP and hormone, they are very low the concentration. This first. Second is it's not compatible like a pesticide, like the common contaminant. So they know they have because of the legislation, the agent, like state federal agents, they don't have enough information. They don't know how to regulate, how to make the regulation. So third is another issue. Another question is because most of this compound is not a toxic, acute toxin, but maybe chronic. So this need a lot of information, research information to support to do this. So that's why I think that this kind of some manufacturer develop some chemical. So we using, then we find the issue, then go back to ban kind of this system, just like before the DDT ban, also like triclosan, same thing. The issue, we went using it pretty good, but later find that they have persistent environment, then go back to ban it. So that's why our rule just research the parts we want to see, if these can persist in the environment or not. If we, also if persistent in the environment, can we develop some strategy or develop some methods to remove them?
Okay. With that, I think we'll conclude our seminar for today. We will be here afterwards. We're going to take a tour here at the center. If anyone's interested in going on that, you can meet up in the front. I also wanted to mention that our next seminar will be on microplastics in the Great Lakes on October 8th. So if you want to tune in for that or attend here, it will be a webinar from New York. And then also for those who are interested, we have a PPCP discussion group we've organized. So if you want to sign up, there's a sign-up sheet here in the front, or for those listening online, can email me at naholm@illinois.edu. And we'll be having quarterly meetings to discuss some of these issues on pharmaceuticals, and then as well as a conference on April 4th next spring. So thank you all for attending today.